This Week in Virology, the podcast about viruses, the kind that make you sick. This Week in Virology, episode 20 for February 12th, 2009. I'm Vincent Racaniello.
I'm Dick Despommier.
I'm Alan Dove.
And joining us today is Matt Evans. Welcome, Matt.
Hi, good to be here.
Matt's from Mount Sinai School of Medicine here in New York, and he is our expert on Hepatitis C virus, which is going to be our main topic today. But we have a couple of things to chat about just before we do that. Here's the question. Whose birthday is it today?
I think it would be Charles Darwin if I had to just guess.
And you'd be right. How old would he be?
Something like around 200 years, as I recall.
That's right. Happy birthday, Chuck.
That's right. Way to go.
Very busy week. It's Charles Darwin's birthday and Abraham Lincoln's birthday today. And then on Friday at 6:30 p.m., we have 1, 2, 3, 4, 5, 6, 7, 8, 9, 0 day in the Unix timestamp. Then, of course, Saturday's Valentine's Day. Yeah, that's the second since the Unix epoch began in 1970. So the number of seconds will be 1, 2, 3, 4, 5, 6, 7, 8, 9, 0 at about 6:30 this evening. 6:30 Friday evening.
I mean, that's amazing, Dick. Why are you making faces?
I haven't got a clue as to what he's talking about.
It's a number of seconds that have elapsed since Unix time clock was started. And it happens to be 1, 2, 3, 4, 5, 6, 7, 8, 9, 0. Is that right, Alan?
Yes, Friday evening.
Sure, that's cool.
Okay.
So what do you do to celebrate that?
You touch a particularly interesting file, I think.
This is getting awfully geeky, Alan.
Yes.
It is.
Well, as if virology is not, right? Well, anyway, Dick.
Yes.
Ted, how was Ted?
Awesome. Absolutely awesome.
Were there mosquitoes in the jar?
You know, where I was sitting, I thought not, because how could he possibly have done that? But, you know, I couldn't see them because I was in one of the last rows in the downstairs zone of where the Tedsters were all sitting. They're hard to see, obviously. I mean, that's why they can get away with murder. The point is that Bill Gates, he's in the foundation business now. He's not in the Microsoft business anymore. He has to dramatize his plights to get people riveted to the spot, so to speak. So to pretend or to actually release malaria mosquitoes that have the potential for transmitting malaria, not malaria-carrying mosquitoes. It's not a big deal, by the way, because there are a lot of mosquitoes that live in California could already carry malaria. Anopheles freeborni is the one. To say that I'm going to release a few more mosquitoes into the air is no big deal, but if you release it inside of a building, it's like crying fire in a crowded theater. It gets the people on their edge of their seats and a little bit nervous about it. So it did bring a laugh, and then later on you saw all the blogs that said, Gates Foundation releases disease on world, and Ted, you know.
Plenty of virus jokes, too.
Plenty of virus jokes, and,
Sure.
you know, he did stimulate a lot of interest, though, so there's no doubt about that.
Was there some good virology at TED?
There was and there wasn't. There was a fantastic animation of dengue fever entering a cell by a group.
Dengue virus entering a cell, Dick.
Dengue virus that causes dengue fever.
Sorry to correct.
No, that's quite all right. We're a virus show here, guys, and I'll stick to the rules. But we also had another event in the evening. They closed down a whole row of restaurants in Long Beach, and they had 2 singing groups, and the second singing group's name was Dengue Fever.
Great name for a group. I think I've heard that, actually.
And I stuck around for the first group, but I must be honest with you, I didn't last after that, and I had to go to bed after that.
Now, wasn't Joe DeRisi there also?
Yes, and we had a lot of other very high-profile people interested in spreading good and interesting ideas, and I was energized by the whole thing.
Did you mention TWiV at least once?
I mentioned it several times.
Excellent.
In fact, I talked to the people from Google as to whether they were interested in actually connecting up with it, and they said they'd look into it. So maybe they will.
Well, if you Google TWiV, you find us.
Yeah, you're number
That's
2,
right.
I think.
Number 2, excellent. You know what? No, I think number 1 is This Week in Villains.
Same thing, right?
It's a comic book site. That's right. It could very well be.
These are the real villains. Come on.
Okay, Joe DeRisi is a virus hunter, and his talk is already up there on TED. He's at UCSF, and he uses microarrays to detect new viruses. So we'll put a link to that because everyone probably should hear it. It's only 18 minutes. And of course, Dick, when your video is posted, we'll put the whole thing on TWiV.
Excellent.
Alan, do you have a breaking story on rhinovirus?
Yes, I do. I have an interesting story that showed up. As a science journalist, I get these what are called embargoed press releases. So the journals, about a week before the paper is going to come out, they send out the press release privately to the journalists, and we're not supposed to talk about the papers until they actually are published, but this gives time to research the story and actually write something intelligent to come out when the paper is published. So this paper, which is supposed to be on the Science Express website 41 minutes ago, but they apparently haven't put it up yet, is about sequencing all of the known rhinoviruses. So this is Claire Fraser-Liggett, Stephen Liggett, and their the usual suspects from the Venter Institute, University of Maryland genome sequencing group. And if you followed the work that this group has been doing lately, what they'll tend to do is go into some field and just sort of grab everything they can find and sequence it, which is can yield some very interesting results. So in this case, what they did was they went to a repository of all known or the largest available repository of rhinoviruses. And these are clinical specimens that I think have been collected over the past couple of decades, representing 99 known serotypes of rhinoviruses. Now, of course, as virologists, we know that there are a lot more serotypes out there, but we happen to have this collection of at least 99 different serotypes. So this team went and sequenced all of them, and they put together a taxonomic tree of all these known rhinoviruses, and they found that they break down into about 15 major clades. So it's interesting, that you can trace the lineage of different viruses and see the diversity of this group. And they also found some interesting things within the individual sequences. So the rhinovirus has a 5' untranslated region. It turns out there's a lot of variation in that. And as we've seen with poliovirus, you might have, it's reasonable to hypothesize that in rhinovirus you're getting some of the variation in the 5' UTR changing the virulence of the virus. So that may be something that accounts for differences in virulence. They also found some conserved regions, some regions of the virus that are not changed. All right, so they found the variation in the 5' UTR. They found the conserved regions, and there's also a fair amount of evidence for significant recombination events, which I thought was kind of interesting, which suggests that you've had at various times 2 strains of rhinovirus infecting the same person, and you get major chunks of the genomes being exchanged between the viruses, which is probably a major source of genetic diversity in these viruses.
When I was at this meeting in Switzerland a couple of weeks ago, a group presented evidence for recombination in new isolates. And the conventional wisdom with rhino is that you don't get multiple infections. So that was surprising. So apparently there are, and we just haven't been able to detect it before. So there isn't exclusion. Multiple serotypes can infect the same cell. I think we didn't see it before because we weren't looking properly, most likely. But now with these deep sequencing efforts, you can pick it up.
So, Alan, if you don't mind me asking a virology question at the lowest possible level, how big a difference in terms of genomic sequence is there between the most different strains of rhinovirus? How much sequencing work was involved?
Well, let's see. How do we quantify this? There's the paper, as you might imagine, has a fairly complex set of data analyses and a complete. I was really hoping this thing would be on the site so we could all be looking at it at the same time. So they've got this very nice genome tree for all known human rhinovirus serotypes.
There must be a lot of conserved regions too, then, right?
Yes. There are a lot of regions of the virus that seem to be highly conserved, and obviously those would be things that you would want to go after with antivirals, or possibly even vaccines, if you can find antigens that are conserved.
But I was just thinking about the amount of work involved. How long would it take if you had all these serotypes sitting in front of you, and you had a big sequencing machine in front of you, and you had X amount of conserved region? How long would it take you to find out the differences?
They obviously had an entire sequencing center at their disposal. Are you talking about in a clinical setting where you might analyze this from a patient?
Exactly right. I mean, for instance, I gave a presentation on this TWiV once about the West Nile virus. As far as I remember, there were 23 different strains of this virus, and they were all in Vincent Deubel's laboratory in Paris at the Pasteur Institute. And he had sequenced them all, but I think the reason why he could do it is because the biggest difference between the strains of viruses was only about 10% or so. So he only had to sequence 90% was identical. So that means that you only have a little bit to worry about.
No, you still have to sequence it all, though.
You still have to go from end to end, no matter what.
Sure, because
just to make sure that there's no base substitution.
Well, you don't know what's going to be different.
Well, this speaks back to the Joe DeRizzi talk at TED, where he could just use a chip that would be able to analyze all the sequences at the same time.
Right.
Yeah, you could set it up on a chip that would tell you if your isolates are different or not.
Gotcha.
Yeah.
All right.
Right. So what you would do is start with, for clinical use, I would think you would start with something like this tree that they've constructed, and then just encode a DNA chip that's got all of the differences in all those 99 serotypes, and then with a simple swab, you'd get an answer, and you'd say, "Oh, you're serotype 37,"
Right.
that sort of thing.
But Dick, with deep sequencing, for $5,000, you can get about 400,000 to 500,000 bases sequenced. So you could, in theory, give them a mixture of all the serotype RNAs and tell them to sequence the whole thing. So it would cost you $10,000 to have it all done. It's not bad.
$10,000 is doable.
You just do the whole thing, and when it's down to $1,000 or less, that's the end of microarrays. You don't bother. You just sequence everything.
Right.
Wow.
Amazing.
Wow.
So, Alan, I presume they didn't include any of the new Rhino C isolates in this sequencing.
I don't think so. I think this was from just the ones from the existing archive.
Right. Well, I look forward to seeing the data because we work on several rhinos for which there is not complete sequence. I have, for example, a student, Angela, who was on this podcast before, who doesn't know the first 60 bases of the Rhino 1A genome. So this is a way to find it.
Yeah.
Of course, there are other ways to get it, but I wanted to mention just briefly this business with Wakefield and autism that's just hit the news the past week, because we have talked about vaccines and autism before. And in fact, all of this began, the anti-vaccine drive began in 1998 when a fellow by the name of Andrew Wakefield published a story in Lancet where he studied 12 kids and claimed that it was measles, mumps, rubella vaccine that caused their autism. As a direct result of his report, the compliance for MMR has dropped in the U.S. and in the U.K., and measles incidence has gone up, and there have been some deaths. Well, now it turns out that there are suggestions dug up by a journalist, Brian Deere, who thinks all his data were fake.
Yep.
Have you heard about this, Alan?
Wow.
I have. I've actually been following the Ben Goldacre story that is part of it was bound up in this whole thing, and I came across this other bit, little investigative journalism that revealed that in fact Wakefield was in the pay of a law firm that was trying to find or trying to generate evidence linking the MMR vaccine to autism. And Wakefield then studied 12 children who, some of whom it now turns out had symptoms of autism before their vaccination.
Mm-hmm.
But he documented this, and now through some newly disclosed documents, it's suggestive that Wakefield may not exactly have discovered a link so much as maybe imagined one.
Goodness.
We should mention that many, many studies have subsequently shown there's absolutely no connection.
Right. Many, very importantly, many much, much, much larger studies. So
Right.
Analyses of thousands and thousands of patients, and there's just absolutely no link here.
Right.
Well, there was also an indication that some of these patients may have had symptoms even going into this trial.
Right.
So,
Yes.
I mean, doesn't that just...
That's just horrible, yeah. Now, he claims that he's right. Of course, he's not going to say, yeah, I did this, because I'm sure there'll be many, many lawsuits.
Well, one of the ramifications is this recent news of this town in Oregon. I believe it's Ashland, Oregon, in which now up to 28% of the kids have not been vaccinated. That's the highest level of non-vaccinated kids they've ever been able to find in the United States.
Wow.
28%. Average is 13%, by the way. Still high.
Still very
It's unbelievable. And the thing is, it doesn't just apply to MMR now. It goes to every vaccine. Parents get afraid of every one of them, and they don't want a polio vaccine. They don't want this. They don't want any of the bacterial vaccines.
I mean, they're doubting the entire medical profession at every level
It's
now.
just it's not a new thing. I mean, this
No.
began with the Cutter incident.
Well-
I don't know what this guy. In fact, if he did do bad things. I don't know what he was thinking.
The point is that every now and then some rotten apple does show up and spoils the entire bunch. And this might be one of those cases where the people would say, see, I told you, we don't trust these people. That's the reason why this doesn't work. Fraud in science, that's what I'm saying.
Yeah.
Yeah, well, someone needs to go on Oprah and all the big shows now and say, look.
That's right.
Well, I mean, I've seen people on, the Today Show or something.
Sure.
And,
Of course.
and they're just fighting to get that message out there. But,
Well, actually, this is why I now have a huge crush on Amanda Peet. I don't know if
Right.
you've heard about this, but she came out in an interview and said, these parents who don't get their kids vaccinated, they're just relying on everybody else's immunity to keep their kids safe. They're parasites. And she caught an enormous amount of flack for saying this, and I just thought that was wonderful.
You posted about that on Dub Docs, I think.
Yes, I did.
Well, we will put a couple of links to some good articles, and you can read lots about this online, but we thought we should mention it because we have always said on this show that there isn't any evidence for this, and now this makes it even clearer.
So Can I just ask what you think would actually have to happen to change people's minds?
Well, I think the problem at this point is the people who believe in this don't believe in it. They didn't reason themselves into this position.
Right.
I think this has become a faith-based argument, and
I think you're right.
the people who have come to believe it are now identifying with a they've got their support group of other believers, and I don't think that there is a way that you could demonstrate the safety of the vaccine to their satisfaction, because that's now part of their identity, is that this they're campaigning to help their children.
I found my way to some of their websites through these stories that Vincent referred to us, and so these websites are amazing in terms of how adamant and aggressive they are in arguing this point.
Yes, and this is why people like Paul Offit, who've come out very strongly in support of vaccination, have gotten death threats. I mean, it's really the argument has degenerated to the point where these people are just not capable of rational discussion about it anymore.
I suspect that this won't make any difference for the people who feel so strongly, because they will say this is a conspiracy of the pro-vaccine big pharma to discredit Wakefield.
Right. They say that Deere is being paid to discredit it by government and industry research groups.
No, it's really unfortunate.
It's maddening. It's a circular argument that leads nowhere.
We have to get through a few generations, so it's forgotten.
Yeah.
Maybe. Okay, the last thing before we get on to Hep C is we had a visitor in the form of Marburg virus in the U.S. Marburg takes a trip to the U.S., and this is a woman who took a trip to Uganda. She was from Colorado. She went to visit a python cave, which was full of bats. And this was in the end of 2007. And in the beginning of the next year, she got sick. She went home, was treated in the hospital in Colorado. And then, as it turned out, around July, another tourist who had visited the cave died of Marburg hemorrhagic fever. So she went back to the hospital, and yes, she had Marburg, and no one knew it. And in fact, the test just came back last month, a year after she had first visited the hospital. So it's the first confirmed infection by Marburg. Fortunately, she lived because this infection is over 80% lethal.
Why do they call it Marburg, Vince?
Because it was oh, Dick.
I've actually been to Marburg.
Marburg, Germany. First outbreak, 1960s.
It's where Höchst is located.
Höchst, just
right.
the die Höchst. Yeah.
Right.
So Marburg, Frankfurt, and another town that's escaping me. Alan will fill me in here, right?
I don't remember either.
Marburg. All right, let's look it up.
They imported some monkeys for research, as I recall. And,
Marburg, Frankfurt, and
13 workers.
Belgrade.
Which
is not Germany.
No.
It's Yugoslavia. Well, at the time it was Yugoslavia.
Right.
That doesn't exist anymore.
Yep.
But it was actually the first member of that family to be discovered, which is called the filovirus family. Dick, do you know the other member?
Ebola?
Dick wins the prize. So that's the two members, Marburg and Ebola. Terrible hemorrhagic fever. These are RNA viruses. They're enveloped. They have a funny elongated shape, which you can find on many nice pictures at CDC. And as I said, over 80% lethal. So this woman is very lucky. None of the people that cared for her got infected. It often happens in hospitals.
Yeah.
This, we should say, in the German and Yugoslavian infections, the virus was brought there in African green monkeys.
That's right.
Which were going to be used in the lab. So lab people got sick and their contacts got sick. And so this lady's pretty lucky. Now, a woman had traveled with her, and we don't know yet if she's going to be sick or was sick. So they're checking. But, Dick, doesn't this just another example of the world getting smaller, right?
That's right. Someone once said the best vector for dissemination of diseases is a Boeing 707.
Yes.
Or now it would be a 777,
Now
right?
that's right.
With 800 people in it.
Exactly, or an Airbus.
So let's keep an eye on Marburg because, I have a feeling it's been here before and we just didn't know it. And it'll probably come back. Someone wrote an email to me about this and said, well, what do we do if there's an outbreak in the U.S.?
What do we do?
Yeah.
Nothing. You have to just contain the people, I think. There's no antivirals. There's no vaccine. You have to quarantine everyone.
That's right. Bury medicine.
Fortunately, this is one of those infections where it's quite clear when you have
that's right.
the infection. It's not like polio, 1% of infections. So quarantining would work.
And it's also not ridiculously contagious like measles or something. This is, you do have to have a fair amount of contact with somebody to get it, probably.
Right.
I mean, there are strains of Ebola that are like Ebola restin that's very contagious.
But that restin is cool because it doesn't cause disease, as far as we can tell.
Now, does it prevent infection with the more serious?
Of Ebola? Yeah, I would guess so. But not Marburg. I think Marburg is too diverged. Yeah. There was one other thing I wanted to mention about this. So the problem with these viruses, we're not really sure what the reservoir is.
Could be bats.
It probably is bats, but I read about an experiment CDC had done. They went to a cave in Africa, I don't remember the country, and they put test animals throughout the cave, and they never got infected. They came back at another rodents of some kind. They didn't get infected, which doesn't mean a lot because it's a hit and miss proposition to be infected by a bat. But we're going to have to work on this a bit more, but it's so dangerous. You need to do it in BSL-4 facilities.
So this woman caught it by inhalation, do you think?
Well, she didn't say she touched a bat.
No. Because you can get rabies like that.
Yeah, in the cave, you would get an aerosol, right?
That's right. Inhalation rabies is a very fulminating infection.
Is it a good idea to let people travel in bat caves, Dick? No.
Well, ask the people at Carlsbad Caverns.
Well, Uganda has shut down that cave now.
Yeah, they have shut it down.
Yeah. I mean, if I were Craig Venter and I wanted to do the most good in the least amount of time, I would collect every species of bat, and I would want to know what the natural history of the virus infections for those bats were. I want to see all of them. Remember I said a fifth of all the mammals on the planet are bats.
It's amazing.
So we watch this space because we're going to see more.
Dick, the problem with that is Craig wouldn't get to use his boat to do that.
This is all true.
Craig, if you're listening, I'm just yanking your chain. It's fine.
Yeah, he's not going to fund you anymore. Before we go any further, we have to listen to a brief announcement from the wind. Now, I have to apologize for all this noise in the background. We have a windy day here in New York, and it's wreaking havoc on my studio. We have a brief announcement from ASM, the American Society for Microbiology. ASM is been very helpful by putting our podcast on the front page of microbeworld.org, which is their site where they publicize all their podcasts. And Chris Kondayan has been a great help there at ASM, and he asked if we could plug their annual meeting, so we'll do that.
On May 17th through the 21st at the Pennsylvania Convention Center in Philadelphia, the American Society for Microbiology will hold its 109th General Meeting, the largest annual gathering of microbiologists in the world. Visit the General Meeting website at gm.asm.org to view the preliminary program, register for the meeting, or reserve your hotel stay. That's gm.asm.org.
By the way, the other virologist at the TED meeting, I just thought of his name, is David Belinsky.
Mm-hmm. David
Belinsky actually is not a virologist. He's a trained medical illustrator. But what he did was he animated the entrance of the dengue fever virus into cells and then showed why it escaped from the vacuole by causing a fusion of the two sides of the same membrane together, which creates a hole actually in the membrane, and out goes the genome into the cytoplasm.
I've heard of that kind of thing, yeah.
I think that's a great segue. Absolutely right.
Actually, I on that note, so he actually works for a company called XVivo, or
exactly.
XVIVO.
That's correct.
And they have a lot of amazing stuff on their webpage, including that movie. Should be on there. If it's not on there, they're very similar, if not the same movies on, molecularmovies.com, I think.
Ex vivo.
Ex Vivo.
All
It's
right.
a really great company.
We'll put a link to that. But Dick, did he have the virus for a live sitting when he did that?
Yeah, this little jar and out came the deck. No. No, he didn't go that far. But he was absolutely entertaining and educational at the same time.
Now we thought we would talk about hepatitis C, and that's why we have Matt here.
Hello?
And Matt is a professor at Mount Sinai here in New York, who has worked on hepatitis C his entire scientific career. Is that fair to say?
Since probably halfway through grad school.
Halfway through. Oh, you started on something else.
I started on retroviruses with our neighbor here, Steve Goff.
Yeah, so Matt was a graduate student with my colleague Steve Goff, who's right next to me, then went on to do a postdoc with Charlie Rice at Rockefeller.
That's right.
Now he has his own lab at Mount Sinai, and he actually has a wonderful paper this week in Nature, and it would be a good time for him to join us. And so here he is, Matt.
Happy to be here.
And your paper is called Human Occludin is a Hepatitis C virus entry factor required for infection of mouse cells.
Right. And just to be clear, that's I'm a co-first author on that. The actual real first author is Alex Ploss, and he's a terrific postdoc who's still in the Rice Lab. This paper is out of the Rice Lab. So.
This is work that you participated in when you were there.
That's right.
But clearly you're going to take advantage of it, right?
Both of my big stories from the Rice Lab were co-authorships with two different German postdocs who were there at different times, and those were just wonderful collaborations.
It makes science go better when you collaborate.
It makes it go a lot faster and a lot smoother.
Yeah, but
Exactly.
the problem is that we have big egos in science, and often we don't want to share.
Right, absolutely.
So we have never talked very much about Hep C on TWiV, so why don't you tell us some salient, important stuff for all our listeners?
Sure. Well, you mentioned it a little bit when you talked about the last 100 years of great virology, or last history of great virology, but I mean, you touched on one of the biggest points is that it was an interesting virus because it was actually cloned before they could actually culture it or really study it in any detail. But for I think from the mid 70s, Harvey Alter had done a lot of great work to show that there was a non-A, non-B hepatitis that was probably responsible for the majority of liver disease. In fact, at one point, I think they said it was probably about 10% of transfusions resulted in this non-A, non-B hepatitis. So there was something very prevalent out there that we couldn't screen for because we had no idea what to actually try to identify. And you gave Michael Houghton credit, which he really deserves for him and his group at Chiron actually identifying the genome. They did some really heroic screening measures to identify a piece of the nucleic acid that is the HCV genome that they then extended to actually clone the full-length sequence. And that was, they actually, he has a couple of reviews out that talk about this saga, and they did an amazing number of failed experiments before they actually got something to work, and what they got to work was still heroic. They basically had a expression library from, a cDNA expression library from an infected chimp, all the RNA from that chimp and it's cloned into this. And they screened it with antibodies from also an infected chimp that they thought were reactive against whatever this non-A, non-B hepatitis was.
What had that chimp been infected with? Serum from
Serum from a patient, absolutely. So they could transmit the virus in chimps really, really well. They knew it was there and it was working. They just didn't know what it was.
So probably today you would just sequence the serum of the chimp.
Well, you wouldn't know what to amplify precisely. I think, again, maybe that DNA chip would be the best thing to do is apply this on there and at least find out what kind of virus it is. So
if you were to do that, would it cross-react with flavivirus sequences?
There should be some regions. I mean, it's still quite different. It's its own genus. So HCV is in the family Flaviviridae, but it's its own genus, and it's about 7 different genotypes within that genus. And so there's a lot of diversity within the HCV family. The problem was after they isolated the full-length genome, we couldn't actually do much with it. They could take it and then try to put it in cell culture or in chimps, and it wouldn't actually do anything, even in a chimpanzee. And so what subsequently was determined is that there was a little piece of it missing, and I think Charlie Rice had a lot to do with identifying the far 3' end of this genome, and it's a little difficult to find. It was after this long, repetitive stretch of U's and C's in this RNA genome. And it coded for this conserved RNA structure that's important for RNA replication. So once Charlie found that and added it to this, the Michael Houghton genome, they could put this RNA into a chimp's liver, by just injecting it, and the chip got sick. So that was a huge advance at that point.
So that was a collaboration between Charlie and Houghton? Or
It was separate at that point, but it's as far as I know.
So Charlie found
I wasn't
the
involved
sequence.
in the field back then. But
Charlie found the sequence, and Houghton...
he extended the sequence.
He saw the paper and made...
So Charlie, Steve Feinstein, and Alexander Kulikov at that point in Charlie's lab had just continued the work to identify the rest of the genome. Yeah, I think that was probably the real starting point. I should say that just identifying any piece of the genome was a huge advance because they could then develop an assay. Chiron developed an assay to be able to screen for this virus. And that effectively stopped a huge route of transmission. So before that, any blood product, like treatments for hemophilia or blood transfusion, could often result in transmission of this virus. Wow. And just developing an assay technique to screen that out was a huge public health advance.
So how common is it in the blood bank?
Actually, no, I'm not sure about the numbers. I mean, before that, they were saying 10% of transfusions would result in this. So, but I think transfusions are often, multi-donor instances.
That's true.
So,
You're right.
So today there's still a lot of Hep C, despite screening the blood supply. So how is that transmitted?
So a large proportion of that are old cases. When you're first infected with this virus, there are almost no symptoms, and so you never know when you're infected. It's about 20 years later when you start to develop some sort of liver condition that people usually pick up on this. And so what we have now is a huge wave of patients who were infected in the 70s and the 80s that are now coming down with liver disorders that need to be treated. So I think we're probably, we haven't hit the peak of that yet. And even without fresh transmission, there's still a huge public health consequence for this.
But there is still transmission, right?
There is. A lot of it is probably either in developed countries, it's probably through intravenous drug use.
Right.
And then in less developed countries, it's still a medical problem where we're not sterilizing needles and things are causing massive transmission.
Is the liver the only organ that's infected?
That's a controversy. I think most people would say that it's only hepatocytes. And we're not exactly sure even how many hepatocytes in a person are infected. Our detection methods aren't that good. But we know hepatocytes can be infected. There's a lot of controversy about whether or not there's neurological tissue that can be infected or immune cells. And I think at this point, I would say no. But I think it's still a wide open question.
And this remains a fairly hard virus to work on, right?
It is. And I'll be complaining about that a little bit today. So at that point, they had a genome. They could put into a chimp and get it to infect the chimp. But, the easiest way to work with a virus is in tissue culture. You can really study, all aspects of its replication if you can actually get it to replicate in a cell. Unfortunately, no matter which genome people tried, I think you mentioned before they had tried certain serotypes or whatever. It turns out people had tried every isolate they could get their hands on from every patient by transfecting that into cells and culture and looking for any evidence of replication. And for the most part, there was almost, a complete failure in doing that. A huge advance came in, I think it was 1999 by Ralph Bartenschlager's group in Germany, where one of his postdocs, Volker Lohmann, constructed a subgenomic replicon, an HCV genome, where they took out the structural proteins, which probably aren't required for RNA replication inside a cell, and replaced it with a drug-selectable marker. And they could use that selectable marker to isolate rare, successful, sustained replication events in cell culture. So subgenomic replicons aren't new, but this was the first time they got one to work with HCV. What they did was they took these RNAs encoding this replicon and transfected it into this one special cell line, HUH7 cells, and it's a hepatocellular carcinoma-derived cell line. And they could observe some replication events. I think for every 10 micrograms of RNA they put into 6 million cells, they got maybe 5 to 10 replication events that they could isolate. So it happened, but it was incredibly very rare.
And that's why they couldn't see it previously.
Right,
it was so
right.
rare.
If you put that genome in without a selectable marker, you never
it.
observe it.
So that was not serotype specific?
That was, they used a genome that is particularly good at it, it turns out. I mean, this wasn't an accident. I think they went through a large number of them. And they got some replication events. It was then actually people again from Charlie Rice's lab that figured out what was going on that allowed those rare events to occur. And there were a couple things. The genomes that were replicating had mutations in them that made them better at replicating in these cells. So we called those at that time adaptive mutations. And there also was something on the cellular side where those cells that happened to be transfected with those adapted genomes who are often better at supporting RNA replication. And they could show that by curing them of virus with interferon and then retesting new genomes. Actually, maybe it would be worth mentioning that, one of the problems with HCV is that there aren't very good therapeutics to actually target this virus. There's the only prescribed therapy right now is interferon based, which is associated with a huge number of side effects, and you have to take it for months before you even know if it's working or not. And in fact, it only works in genotype 1, which is the major genotype in Western Europe, and here in the U.S., it only works about half the time. So your patient suffers for often no reason. In other genotypes across the world, like genotype 2 or 3, it's probably 80% effective. But then there's still a large failure rate for very expensive, very deleterious treatment.
So what's the outcome if you have Hep C infection?
Nothing initially. At least that's what I think, or what a lot of people think. And then after 10 to 30 years, so I should say you're first infected, you have a chance of clearing the virus immediately. And that appears to be associated with a multivalent, strong immune response. But if you don't, only probably 15 to 20% of people initially clear the virus. The rest of these people develop a chronic infection that lasts for the rest of their lives if it's not treated. Of those, the rest of those infected patients, I'd say something like 70% of them go on to develop chronic liver disease, which can be cirrhosis or liver dysfunction. But another 20% of this group will actually go on to develop massive liver failure, which actually probably requires some sort of transplantation to deal with it. 1 to 5% of infected individuals actually develop liver cancer, which is the worst possible consequence of this virus.
So it shares some features with hepatitis B then?
Absolutely. I think we often are following the same paths of investigation.
But that one replicates much easier in liver?
It really does. Yeah, the systems are at least a little better. Not great, though, for that virus either.
So if you had to make a guess here in terms of evolution, because this is the 200th year of Darwin.
Yes.
How close is hepatitis C to being adapted to human rather than hepatitis B, if you had to compare the two?
Well, first of all, they're totally different viruses. One's a DNA virus. Hepatitis B is an HCV is an RNA virus. So these things are called hepatitis viruses only because of their clinical symptoms.
But in terms of adaptation to their replication cycle, they show gradations of ability to do this. Would you care to speculate as to why that might be?
No. Actually, I'm sure, I don't think I'm capable of speculating on that.
I mean, it just sounds better adapted. It doesn't cause much disease in the beginning. It lasts for 20 years. You might die from something else in between. So that there doesn't appear to be a lot of selective pressure to...
Well, I think most viruses are not directly causing pathogen, or that's probably a sweeping statement. Viruses in their natural host probably aren't going to be causing a lot of damage. That's what you were going for?
That's
Great.
Exactly what I was
I'm glad I can play ball.
Sure. Like bat virus, like rabies.
Well, it depends. It depends on their life cycle, though. I mean, if you have something like dengue virus, there's actually a selective pressure to replicate to the highest possible population in the bloodstream, so you'll get into a mosquito for your next host.
Oh, absolutely.
Yeah, but there's no reservoir host for that. There might be for this.
Well, we know that so far you can only infect chimps and humans. So it's probably...
Has it been found in any animals in the wild?
Actually, I don't know. Maybe I should figure that out.
Because there are animal hepatitis
you said
C
it's
viruses.
a flavivirus.
Yes.
Now, is this one of the few flaviviruses that are not vector-borne?
Gee,
well, that's why it's in its own genus, right? Hepacivir.
I mean, when you told me this just now, I was unaware of the fact that there were other flavivirus members that were not vector transmitted.
Well, I guess it's not technically a flavivirus. It's in the family Flaviviridae.
I'm not a virologist. I can say that.
He's right. It's in the family, but it's its own genus, and it's not vector-borne. Yeah, but there are animal hepatitis B viruses.
Sure,
but we don't know of any.
There's woodchuck hepatitis B virus.
Exactly, and they're in duck and others, squirrel. But there are no animal hepatitis C viruses, which would be interesting for someone to go out there.
We could really use them actually
and find
to study and So there's not a lot of options once you're infected with HCV. Again, there's just this interferon treatment, and that only works part of the time. But there are actually some really good drugs on the horizon. I think Saul was talking about this in
yes.
a couple TWiVs ago. I think he was a little optimistic about how close they are to actually being on the horizon. Probably the first compound will be out. That's Talabavir from Vertex, and that's in phase 3 right now, but that could still be 5 years away.
What is that targeting?
That's targeting a viral protease, the main protease of HCV called NS3. And it's, so that's an obvious target for antiviral development. You can actually take that protease and do protease assays in a test tube. So you can screen a lot of compounds very easily. So they could start screening for those kind of inhibitors before there was even a replication system in cell culture. There are also, there's an RNA-dependent RNA polymerase in HCV, and so that's also a very good target for drug development. And there are certainly companies that have compounds, I think, in phase 1 and phase 2 trials right now to try to look at those. The problem is, and it's an obvious problem, and it's already been shown in cell culture, that the virus can easily mutate around any one of these inhibitors. And so it's one of those instances, again, where you're going to have combinations and overlapping combinations of things that don't have the same resistance profiles. And even separate protease inhibitors so far seem to have very similar resistance profiles. So this is going to be a problem in terms of targets.
So I'm intrigued by the term occludin. This protein is called occludin, and yet it helps the virus get inside, or it keeps it from getting inside.
That was named.
Does that work?
That was named for its cellular role. It's it's the protein that forms a tight junction in a polarized epithelial cell, so it occludes the epithelial space.
That's an unfortunate double entendre.
Sure, absolutely.
So tell us why you went after occludin and claudin and.
So when I started in actually looking at HCV entry. It was almost because we knew there was a problem in identifying what factors were required on a host cell. A lot of people were interested in finding these receptors, or I like to actually think of them as entry factors, because receptors have a certain connotation associated with them. You have to show they're actually directly interacting with the virus on the way in. I'd rather say, what does the cell actually need to support entry of this virus, whether or not it's direct or indirect? And so we knew that you could only really see HCV entry in a cell of hepatocyte origin. And at this point, we have a couple systems that let us actually study infections in cell culture. There's this amazing Japanese fulminant hepatitis 1 isolate of the virus. It's a genotype 2A virus that they isolated from a patient with fulminant or actual, acute high-level liver damage that may or may not have been associated with HCV. But they got this genome out of the patient that, for whatever reason, makes particles in cell culture. So this was work from Barnschlager again and Thomas Piechman, Brett Lindenbach and Charlie Rice and Frank Cesari. There were a flurry of papers coming out at the same time, where this system that we could actually now study the entire life cycle in cell culture.
How many years ago was that now?
That was in 2005.
Okay.
So that was actually an amazing time to be in the field, because all of a sudden all these projects we wanted to actually do were possible.
That's just when you went to Charlie's lab also, right?
Yeah, so I was there. I started there in 2003 or something, so I got to see the explosion of that system. That was amazing.
Bad news for the patient and good news for science.
Mm-hmm.
Well, yeah, I mean, so thankfully it was one patient, and
Shout out to Helen Lane.
Yeah.
Yes.
Henrietta Lacks. Helen Lane was an alias.
I didn't want to use a real name.
So the problem is, this is one genotype of the virus of thousands of isolates that have been identified. There's something very special about it, but at least it worked.
Can you take pieces of it and put it into other genotypes
Right,
and get them
right.
to
So,
grow like crazy?
Absolutely. There's a way to track down the important pieces, and the way they've done it is take another genotype 2A strain of the virus that's relatively close related to it and keep swapping pieces. And what they came down to, I think, and I'd have to look it up to be sure, is a piece of the NS3 protein, this helicase protease protein, and a piece of the RNA-dependent RNA polymerase seem to be most important. Also, maybe a piece of the 3' untranslated region as well. But so they've gotten down to very small pieces that seem to support the ability of this other genome to make particles. The question is, what is the mechanism, and that's still unclear. But it's a fascinating development that's allowed a lot of other work to be done as well.
So can you, under what conditions do you have to work with Hep C? It's not airborne transmitted.
It's BSL-2 plus in the U.S. It's BSL-3 in Europe. Which BSL-2 plus just means a normal tissue culture hood, but you disinfect everything that comes out of that hood.
Okay.
Right? So it's a little safe. I mean, you really do have to inject this virus to be infected by it, and even medical exposure, accidental medical exposure, where you have a needle stick with somebody who's infected, often does not result in infection. So it's not highly contagious by that level, which helps us actually work with it in the lab a little easier, I suppose. There's another system we've been working with, which are lentiviral pseudoparticles, where you can actually take basically a lentiviral particle that only expresses a reporter gene and stick the HCV protein envelope proteins on top of it. And those envelope proteins give that particle what appears to be HCV tropism. And that's a great system because it allows us to study entry in a cell that isn't good at supporting HCV RNA replication. And really only hepatocytes can support HCV RNA replication, so we would be rather limited without that system. Sneeze. So we're jumping around a lot here, but I hope that's okay.
Oh, it's great.
Does anyone mind?
I don't mind.
All right.
No.
So when I joined the Rice Lab, there were two systems that were just developed that allowed us to actually study the entry of this virus into a host cell. This authentic HCV virus made from the JFH1 genome and pseudoparticles. And what we did was survey a lot of cell types to try to find out which cells could support just entry of the virus and which ones couldn't. And it was only hepatocyte-derived cells that were capable of doing this. And furthermore, it was only human cells that were able to do it. So we knew that there was something very special about what was on these human hepatocytes. And so our goal was to try to identify what those factors were. And the way we chose to do it was to take a non-hepatocyte cell, in this case a 293 T cell, which is a kidney-derived cell line, and make sure it expressed all the factors we knew about at that time. There were two factors, CD81 and scavenger receptor, which we thought were HCV receptors. And we made sure that 293 T cells had a lot of both of those, and we said, okay, what's missing? So we delivered a cDNA library from an hepatocyte cell and screened for cDNAs that could render those cells infectable. And to make this long story short, we found Claudin-1 was, when expressed in a 293 T cell, able to render that cell able to support entry. Furthermore, a number of other non-hepatocyte cells were able to support entry when we expressed Claudin-1 in them, along with the other HCV entry factors. And so we were able to, for the first time, infect in a non-hepatocyte cell with an HCV
And this
species.
is just delivering the lenti reporter.
Well, we could do it with the pseudoparticle system, but we could also do it at a very inefficient level with the JFH1 virus. So we knew at that point the difference in ability of 293 T cells and HUH7 cells in supporting RNA replication, and it looked like the inefficiency of entry of infection with the cell culture virus was due to replication and not an entry defect. So expressing the Claudin-1 in those cells made them as good at supporting entry as the hepatocyte cell lines. We did other experiments where we silenced Claudin-1 in the hepatocyte cells that were naturally infectable, and it would prevent infection of those cells. So that was good back and forth evidence that this was an essential entry factor, I prefer to say, at this time. So we went ahead and studied its mechanism a little more, but all we know is that it seems to work after the virus is attached to a host cell, and somewhere just before the virus is endocytized into the cell to then fuse in an endosome and infect the cell. So it's a post-binding HCV entry factor.
So did you try that in mouse cells at all?
We did. We tried to express the three factors we knew at that point, CD81, scavenger receptor, and Claudin-1 in mouse cells, all the human versions, and we weren't able to render those cells able to support entry. And so that actually leads to our most recent paper with Alex Ploss, where we took a mouse cell that we had engineered to overexpress the human versions of Claudin-1, CD81, and scavenger receptor, and screened for human hepatocyte cDNAs that could render that mouse cell now able to support entry. So it's doing almost essentially the same approach. We're just doing a cDNA library screen where we're hoping to make a cell able to support HCV entry. But in this case, we're being a little more aggressive. We're trying to make it work in a mouse cell. And we were surprised, I have to admit, when it actually worked. And we were able to find a fourth entry factor that, when expressed in mouse cells, was able to make those cells infectable. So this factor was occludin. It's funny, occludin and claudin are actually both essential components of tight junctions. Those are junctions that seal the basal and the basolateral and apical spaces of a polarized cell. They actually form a barrier between those surfaces. And they restrict, or they help to restrict either soluble species from flowing between these epithelial layers, and they also allow the different membranes to specialize a little bit in their functions.
Are these accessible on the surfaces of cells?
No,
they shouldn't. So they're not actually binding proteins,
well,
right?
There's that's actually the first question that people throw at us when we say it's a tight junction protein, and it's puzzling. Why would a pathogen want to interact with? If it's interacting with these, we're not sure about that. But why would it want to utilize a protein that's in a difficult area to access? There's actually a really interesting paper from Carolyn Cohn, who's now a researcher in UPitt, but she as a postdoc in Jeff Bergelson's lab. She studied how Coxsackie B viruses enter cells, and what they actually use are two different receptors. One's present on the apical surface of a cell, and the other's in a tight junction. And what she found is a progressive interaction with the virus in those cells, where it first binds to the apical exposed surface of the cell, and several intracellular signals occur that actually open up the tight junctions, disrupt their restricted ability. And this virus initial receptor complex actually translocates across the cell surface. She showed this visually, and it's a really beautiful time lapse of immunofluorescence, where the virus receptor, initial receptor complex actually moves across the cell surface into the tight junction region, where after that it's endositized. So we think that maybe these are difficult things for viruses to actually come in contact with, but they might actually be useful once they can get there. Maybe there's something about how these proteins are recycled from the cell surface that the virus can actually jump on to gain access into that cell. Maybe there's some endocytic pathways that these proteins are particularly good at doing that it wants to get in touch with.
So do you think these 4 proteins now are all that are required for entry, or is this
well, no, absolutely not. I think they're the only proteins that really differ between cell types that are required for entry. We think there's a whole lot of other machinery that's absolutely essential, but it's almost invisible because it's expressed in all these cells. Certainly clathrin is essential, because that's how this virus is endositized, right. A clathrin-dependent mechanism. And acidification of the endosome is also required, so factors involved in that are also important. But what's different between cells is primarily localized in these 4 factors. I should say that scavenger receptor and claudin-1 actually work fine whether or not they're from the human or the mouse origin. And it's coincidental that those are the 2 factors that are most liver-specific in their expression. So we think that overlapping high-level expression of both CD80, claudin-1 and scavenger receptor in hepatocytes is the reason why hepatocytes are particularly good at supporting HCV entry processes. But occludin and CD81 are actually very species-specific. We've showed that in this recent paper that mouse occludin only works, barely works compared to human occludin. And so there's a big difference between their ability to support this entry process. So we think that CD81 and occludin, although not responsible for the tissue tropism of the virus, are responsible for the species tropism. So it's nice how they're kind of segregated in those 2 groups.
So you mentioned in the paper that you think that beyond entry, you're going to have to adapt for replication or find factors that the virus requires. Do you know already that in mouse cells the genomes don't replicate well?
Right. So if we can make entry possible in a mouse, would that give us a mouse model? Probably not. So we know, we can do these techniques where you take subgenomic replicons again and transfect those into mouse cells, and very, very, very rarely you can see some sustained RNA replication. And so it's possible in that environment, but it's certainly not efficient enough to make a model around it. So what we envision is maybe trying to select a virus variant that can actually replicate better in mouse cells, or maybe do cDNA library screens in mouse cells to find missing human factors, and then use that in addition to transgenic mice expressing human occludin and human CD81 to maybe throw a mouse model together. But that's that's certainly not a close goal.
But you would like to do that because you want a mouse model, because that's great for the antiviral, it's for studying pathogenesis and everything.
Yeah, chimps are a terrible animal model. I mean, they're for ethical and financial reasons, you just can't do studies in chimps.
And for physical reasons, it's just...
Yeah, absolutely. I mean, they're almost people, so...
So is that something you want to do in your lab? You want to look at the replication in mouse cells, make a transgenic mouse as well?
I think my bread and butter is going to be exploring the entry process, maybe not just of HCV, but other viruses as well. We have a good screening technique. We might as well try to apply it. I can't ignore the idea of trying to pursue this mouse model thing. I think Alex, my co-author, is certainly pursuing the transgenic approach. So I'm more interested in trying to figure out what's missing in a mouse cell to support RNA replication.
In the world of viruses, just to expand this topic out now and to try to give a broader view of this, how many infectious agents, including viruses, and I can think of one very important non-viral agent like malaria, that targets the hepatocyte? You have a lot of those cells in your body, obviously. So you can afford to throw a few of them away, but this gets out of control, obviously.
What's funny about malaria is that it invades cells. Yeah. It invades hepatocytes, and it appears to require CD81, a scavenger receptor, 2 HCV receptors to actually do that process. We don't know if other factors are also required for that. But it's a very interesting overlap.
It would be horrific to learn that malaria is dragging in the virus with it, but
there are plenty of people who are infected with viruses who don't have malaria.
This is all true. Of your list of agents that can infect liver cells, besides the one you're studying now, what would be the other ones? Besides, I mean, there's hepatitis
A, B, C, D. Yeah,
how
absolutely.
many do you think there are?
I think that it's going to be endless. I mean, it's an easy target in the circulatory system because everything goes through the liver.
That's right.
So there's probably going to be an endless number of pathogens that we can identify that go through that.
Somehow, most of us seem to keep them out because we seem to be living fairly healthy lives right now.
Well, I mean, if you're exposed to the wrong thing, most people have a problem keeping it out. It's for HCV, 80% of the people, very healthy people, are still chronically infected with this virus. So there's, if you're unlucky enough to be exposed to the right pathogen, it's going to get in probably.
So one approach that you mentioned is to look for proteins that the human virus requires in mouse cells. Try to perhaps select for viruses that can grow better in the mouse cells.
Sure.
How about the other approach, to go out in the wild and look for related viruses in mice, voles, other rodents?
Absolutely. I think that would be incredible. There are some related viruses. They're not close enough. Mm-hmm. Dengue viruses, all the Flaviviridae have the same genomic structure and very similar replication cycles. And people have tried to make chimeras, at least make things that look more HCV-like, and they failed for the most part in those kind of experiments. But you're right. I mean, there's certainly surrogate models to work with HCV, to study the HCV processes in.
Well, you've just started your lab, right?
I started in July of last year. So I spent... Thanks a lot. I spent probably half that time ordering things. It's amazing.
Of course.
Yeah.
I remember that.
Selecting the color for your walls. And
I mean, how do you know which model of -80 freezer to get? So
call Steve and ask him.
Exactly.
That's right.
He might not even know.
Yeah.
Well, I hope you get your office quiet so that maybe you could Skype in the future and join us again.
Maybe TWiV will get me some good postdoc applicants.
Yeah, well, listen. This is a good
absolutely.
question. So, what would you suggest for someone just going into virology and wanting to do it as a career? You got any words of advice for them?
Work with a really good virologist. I think I was very lucky to have great mentors. I mean, Steve Goff and Charlie Rice are certainly some of the best. And then to be exposed to Vince and Saul and Jeremy when he was here was tremendous in my education. I think that's the most important thing. And those people also attract the best students and the best postdocs. And so you end up being in an environment that you can just thrive in.
Well, we really appreciate your coming up here and spending time. It was great. I think I learned a lot.
Yeah, definitely.
I
cool
learned.
stuff.
Wow,
And
it's a lot of fun.
I hope you return. I just want to. We're long here, but I just want to read 2 emails which are relevant to today's discussion. Okay, then we'll get on to our picks and wrap up this windy day in New York City. So we have one from Joe. Joe writes, "Dear TWiV, just wanted to write and say thank you for the marvelous podcast. I recently finished a PhD on HCV.
Oh, wow.
And I'm just setting out on a career as a research virologist. Your podcast helps me keep on top of news and current opinions. I especially like the background you give to every story. Very good for helping me think about what I do in a broader context.
Wow.
I've even been downloading Professor Racaniello's virology lectures to remind myself of all the basic stuff I learned as an undergrad."
Outstanding.
So that's Joe, and he says, "P.S. If you ever want a TWiV discussion on HCV, I'd love to chip in."
Well, I know who Joe is. Hi, Joe. How's it going? Yeah.
Joe will obviously chip in the next time this is broadcast.
Well, I told Joe I'd make a list of people who would love to join us.
Yeah, Joe actually studies HCV entries, so I'm sure he can correct all the errors I just put out there.
No, that'd be great. We'd like to have as many virologists as possible to contribute to the discussion. And one other email, Dick, I saved this one for you, and this is from Sergio. "Hello, guys. I discovered your podcast a couple of weeks ago, and I've been getting updated from the very first episode to the last one. I really appreciate the work you're doing. The podcast is very informative, very original. The music is great, and very understandable for someone like me. It's not much music.
Oh, well.
I basically work with plant-related bacteria in my PhD research, but at the same time I am very interested in plant viruses. Since in the last episode you mentioned that some very important seminal moments in virology were related to plant virus, I thought it would be interesting to have an episode. I personally think they are very underestimated and underinvestigated in developing countries.
Very true.
Yeah, I think we should do a plant virus episode. I have to find a plant virus expert.
They create a lot of food shortage.
Yeah.
Here's the question. In the last few weeks, my country, Bolivia, has declared emergency due to an epidemic of dengue fever, and there are estimates that the morbidity and mortality will be greater this year than past years. Dengue appears so often in Bolivia after summer floods every year that I wondered why it cannot be controlled in a better way. Right now, the only measure is fumigation with insecticides, but I would like to know what you think about it. Thank you very much, and congratulations on your podcast.
Window screens, window screens, window screens.
And empty all those incidental containers.
Yeah, you don't want the standing water, right there.
No standing water. It's the same control measure you'd use for yellow fever.
Mm-hmm.
It's what allowed the Panama Canal to finally get completed once they found out which mosquito was transmitting it.
Mm-hmm.
They were able to control it because they knew where it bred. And the same is true here. It requires everybody doing their fair share to make sure all of those filled up containers are now emptied out. It's transmitted vertically as well as horizontally, so that you can't outbreed this from the mosquito populations. You have to control their numbers to get this.
So what's the mosquito in Bolivia that dengue is in?
Probably is in the Aedes aegypti.
Aegypti. Pesticides, not a good idea.
Well, if you sprayed it as a residual spray on the windowsills in your houses on the inside, so that when they take the blood meal and they go over there and rest for a minute, they die, that's a control measure for malaria. I'm not sure it works very well for Aedes aegypti, to be honest with you. It's a day biter. You would be encountering this in your daily work and probably not in your house at that point.
Okay, there you go, Sergio, the answer from the expert. If you have more questions, just send them to TWiV at twiv.tv. All right, so that brings us to our picks of the week, science blog of the week. It's called Not Exactly Rocket Science. This is by Ed Yong, who's a science writer in London, and he says it's his attempt to make the latest scientific discoveries interesting to everyone by beating jargon, confusion, and elitism with the stick of good writing.
Wow.
That's quite a metaphor.
Wow.
I thought you would like that, Alan.
Yes.
He has a really good writer, and he's chimed in on a couple of topics lately with respect to microbes that have been good. So check it out. We'll put a link in the show notes. Science podcast of the week is called Neuropod, which Alan probably knows about. It's a Nature podcast about neuroscience and being interested in neurotropic viruses. I like this. I like to understand what I'm destroying with my viruses. So it's a typical Nature podcast. It's slick and well done, but it's good, very interesting. And I listen to it in the car and learn about my brain. And finally, Science Book of the Week. Here's a good one, guys. It's called Phage and the Origins of Molecular Biology. Do you know this book, Dick?
I don't, but you'll tell me about it now.
And Alan, neither do you?
I don't know about this one, no.
Okay, so I read this as a grad student. It was published in 1960. And it's basically a series of articles by the big guys in the field at the time, who are mostly phage and bacteria guys, on the occasion of Max Delbruck's 60th birthday.
Oh, nice.
Sure.
And he was the founder of really molecular biology. He was the first guy to do a single step with bacteriophage, and he and Luria figured out fluctuation analysis. So these are like personal stories about molecular biology. And this book actually was just reissued. It's published by Cold Spring Harbor, and it just came out. It's $29 on Amazon.
Well, your copy is in several pieces right now.
Mine's breaking apart here.
It is.
Look at that.
It looks well read.
It's well read. I love this. And I want to read one paragraph here from Luria because it's so neat. So this is a chapter by Salvador Luria, who won the Nobel Prize for fluctuation analysis, which we'll explain in another show. It's called Mutations of Bacteria and Bacteriophage. And he says, Max Delbruck and I met for the first time on December 28, 1940 at a Physical Society meeting in Philadelphia. He was teaching then at Vanderbilt University. After a few hours of conversation and a dinner with Pauli and Plasek, during which the talk was mostly in German, mostly about theoretical physics, mostly above my head, Delbruck and I adjourned to New York for a 48-hour bout of experimentation in my laboratory at the College of Physicians and Surgeons. I had received from Dr. Bronfenbrenner of St. Louis 2 coli phages active on the same host, P28, later called alpha, later called T1, and PC, later called gamma, and later called T2. These are very famous T phages.
Absolutely.
The reason for choosing the names alpha and gamma was that my typewriter had the signs alpha, beta, and gamma. Beta was left out for reasons of symmetry, the common host being called B for bacterium. So I had not known that he worked at College of Physicians and Surgeons. And so all the stories are like that. They tell, these guys talk about what it was like.
They're 48 hours of binge science.
Can you imagine? Yeah. We played out phages for 2 days. Well, you played them out, you go for a drink, and you come back 2 hours later, you have plaques, right?
Right.
Okay, that should do it for another TWiV. I'm now collecting virology news links at delicious.com/TWiV podcast, and it's open to anyone. If you find a virology story, post it there, and we'll look at it, and that'll be the agenda for the show. I want to remind everyone that you can win a free copy of Principles of Virology if you link to TWiV.tv from your website or blog. The instructions are on TWiV.tv. So I am on Twitter, PROFVRR. Alan is Alan Dove. Matt, you probably don't know what Twitter is, do you?
I've heard you talk about it.
Do you have a website where people could learn
I
about?
have a website, absolutely.
All right, we'll put that in the show notes if you don't want to.
No, I don't know it offhand, so you have to look it up.
So people can go and find out what you're doing. Send us your questions, complaints, what you want to hear about TWiV at TWiV.tv. Any last words, gentlemen?
Just that I have a great time. I'm glad to be back home.
Good to have you back, Dick.
Good to have you back, Dick.
Thank you, Alan.
Thanks, Alan.
And Matt.
Thanks for inviting me. I really appreciate it. It's an honor.
a pleasure meeting you.
Thank you.
Thanks a lot. You've been listening to This Week in Virology. We'll be back next week. Another TWiV is viral.